Hello everyone. Today I will be revising an indoor test. Let me shift the camera up until this point. I hope all of you can see me well, and today I will be revising one of the experiments of microbiology, which is the IMViC test. Today we will be revising the indoor test first, which is the first step of the IMViC test. We are talking about the indoor test. Firstly, let's talk about the objectives of the damn test. Let me just indoor test. We have to talk about the objective first. The first objective would be to know if the organism produces tryptophanase or not, to know if the organism is motile or not, to know if the organism degrades cysteine to form hydrogen sulfide or not. Firstly, we have to know if organism makes tryptophanase, if it is tryptophanase. Then secondly, we have is if it is motile or not, and thirdly, we have to check if it degrades cysteine or not. Cysteine or not to form hydrogen sulfide. These are the three objectives of the Indole test, and when we are talking about other things, let me increase the brightness for a bit. And now we have to talk about other things as well. Now, how do we obtain the information about these things? Firstly, we have. Let's talk about the procedure for this first. I like starting with that. Procedure. When we're talking about the procedure, firstly, sim media is required, then bacterial culture is required, and then COVAX reagent is required. And in this process, firstly, the first step is sim media. Sim media is labeled. Sim media is labeled, then sim media is labeled. Sim media is labeled, then each of the organism is inoculated aseptically. After this, incubation is done for 48 hours. 48 hours incubate at room temperature or body temperature, 37 degrees Celsius. Then after 48 hours incubation, we have add 4 to 8 drops of COVAX reagent, and after adding 4 to 8 drops of COVAX reagent, mix. Afterwards, observe shiny red color on the surface. After observing this, we also have to observe black hydrogen sulfide. We have to observe hydrogen sulfide as well, and also motility. An example would be if we use organism E.coli, then it would show a positive result. And if we use some other organism, for example, Klebsiella pneumoniae, morning, then it would show a negative result. Afterwards, we have the observation table. Also, we have the procedures here. Let me search it more easily. Indole test. Search for the Indole test. It determines the ability of an organism to produce indole from the degradation of amino acid tryptophan. Let's check for the images. I think this will show a positive result. See, it's a more realistic taste and tone. Let me use this again. This one is for the desktop. I think that would be an image. What is the picture? I mean, I think there is one. Okay, we know the cherry red equals positive. Let me use some other image. Like this one here. Cherry red is the positive, and if there is no cherry red color, then it is usually a negative one. And let's talk about the components. We have the sim media. What the hell is the sim media? Main requirement for a suitable Indole test medium is that it contains a sufficient amount of tryptophan, which includes tryptophan broth, indole-free peptone, water medium, urea, indole medium tryptophan broth, and then we have the sulfide indole motility medium. This is the required media for this given test. Next question, sulfide indole motility media. Now, let's check about the reaction involved in this. Reaction involved in indole test, and it would be let's check about the DM. This is the reaction involved. First, you have the tryptophan. If tryptophan is produced, then the tryptophan along with water get reduced to indole, pyruvate, and ammonium. And because of this, the color change will be observed if tryptophan enzyme is present. If not, then this reaction will not be observed. Okay, I'll read the theory for once. Tryptophan plus H2O in the presence of tryptophan enzyme gives indole, pyruvic acid, and ammonium. And two of these indoles, along with para-dimethyl aminobenzaldehyde HCl and amyl alcohol, which is the Kovacs reagent, gives rose indole dye or cherry red color. And the coliforms can be differentiated by their ability to produce tryptophanase. And this enzyme hydrolyzes the amino acid tryptophan into indole, pyruvic acid, and ammonia. Pyruvic acid can then be used by the organism in the Krebs cycle, or it can enter glycolysis and be used to synthesize other compounds that the cell needs. The medium employed in Indole test is SIM medium, which includes peptide and necessary growth factors, but enriched with amino acid tryptophan. And the SIM media can also be used to detect the motility of organism and also to see whether it produces sulfide or not. Hydrogen sulfide is formed by anaerobic respiration. In putrefaction, sulfur-containing amino acid like cysteine is broken down by enzyme cysteine. This sulfur is producing pyruvic acid, ammonia, and hydrogen sulfide. Now let's talk about the discussion and conclusion. In this regard, coliforms can be differentiated by their ability to produce enzyme tryptophanase or not. So let's write the points. How do you identify coliform? They produce tryptophanase. The enzyme, they produce the enzyme tryptophanase. And next thing is tryptophanase. This enzyme hydrolyzes the amino acid tryptophan into indole, pyruvic acid, and ammonia. So if it is a coliform, then it should be able to hydrolyze the amino acid. Hydrolyze. What is happening to my spelling? Hydrolyze. Hydrolyze amino acid tryptophan. Hydrolyze tryptophan into indole, pyruvic acid, and ammonia. Tryptophan then pyruvic acid can then be used by organism in TCA cycle. The pyruvic acid thus produced then is used in Krebs cycle, into Krebs cycle, and then we have Krebs cycle, etc., or glycolysis. And now why SIM media is used. Why SIM media? Because it sulfur indole motility, because it has peptone, and then growth factors. It has peptone, growth factors, and also rich in tryptophan. Okay, now we also have the COVAX reagent. We also have this. What is this? COVAX reagent simply means HCl. Hydrogen chloride plus dimethyl amino benzaldehyde and amide alcohol. After incubation, indole is formed. Indole has a pyruvate structure. The H2 group of pyruvate permits condensation with aldehyde in a two-step process that is formed by amine that is insoluble in water, and organisms that break down tryptophan show the result in indole running a test. Otherwise, no color change is actually observed. Now, what happens when the Kovacs reagent is used? We know that indole has a pyrrole structure and it is red in color. Now, CH2 group of the pyrrole, that is of indole, it allows condensation with aldehydes in a two-step process, and the ring is formed by amyl alcohol that is insoluble in water. Hence, organisms that break down tryptophan show the result in indole test because they are easily able to condensate with the aldehyde and thus form a ring. So tryptophan shows the result in indole test, otherwise no color change is observed. And now, a cherry red ring can be observed in sample if it is usually a coliform. For example, E.coli or Escherichia coli, and the agar must be solid, and excessive addition of Kovacs reagent is not be done. This was the indole test. And now we have to remember what are the medias and everything else. Firstly, let me check. Now, if that is true, then we do not have a problem. Let me summarize it once again. Firstly, IMViC test. The first step is indole test, and the objective of the indole test is to check whether the organism produces tryptophan enzyme or not. Secondly, if the organism is motile or not, if it can produce black hydrogen sulfide, that is, if it degrades cysteine or not. And the procedure has various reagents used. Firstly, we have the SIM media, which is the sulfur indole motility media. It is used to check the motility, and if the organism is motile, then we would see the spreading of the organism in the test tube. Otherwise not, it is concentrated. Then we have this, after the SIM media, we have the Kovacs reagent, which is also important. Kovacs reagent simply includes hydrogen chloride, amyl alcohol, and p-dimethyl, what's that? I forgot. It simply includes p-dimethyl aminobenzaldehyde. Now here is the aldehyde, and it can condensate with the CH2 of the pyrrole of the indole, and it will later form a ring. After this, we also have after all this, if a positive result is there, then it would show a cherry red color, and if the result is negative, then such color would not be seen. And the procedure as firstly, SIM media has to be labeled, then each organism needs to be inoculated separately, and then incubation must be done for 48 hours at 37 degrees Celsius. 4 to 8 drops of Kovacs reagent need to be added. Reagent would be mixed. Cherry red color would be observed on the surface. Black hydrogen sulfide precipitate needs to be observed, and afterwards motility is also observed. This is the first one, which is the indole test. Thank you.